It's my pleasure to welcome Ray Hawkins from Fernando's Gonzalez Lab at University of Toronto, who'll be presenting on ReSCU Nets. And I'd like to remind everyone that we are taking questions via the Slack channel. So please join the track-bioinformatics, etc. Slack channel to post your questions there. And at the end of the talk, I will field the questions to Ray. Please welcome Ray to SciPy.
Okay, yeah, so I'm Ray. I'm from the University of Toronto, and today I'm going to be talking to you about my master's project where I've been developing ReSCU Nets, which are recurrent U-Nets for automating the segmentation of multidimensional microscopy data. And if there's two morals that I want you to take away from this story, the first one is that sometimes the best path to automation is actually semi-automation, and the second one is that we need to continue to design things for small data. There is definitely a trend towards big data in the field, but especially in science, when different labs are studying different things, big data sets aren't always readily available. So to give a bit of a background on what we do, we study collective cell behaviors, and that's because these collective cell behaviors are important drivers of development and disease. So if the proper collective cell behaviors didn't happen during development, we wouldn't be here today, and if the wrong ones happen today, we won't be here tomorrow. For example, during embryonic development, here's a video of xenopus neural tube formation, or frogs, if you're not familiar. And in order for the neural tube to form, there is this epithelial sheet that undergoes coordinated apical constriction, and this constriction allows the sheet to fold into the neural tube. Similarly, in cancer metastasis, cells invade tissues by collectively moving together, and this collective movement requires the coordinated polarization of several cytoskeletal components. Now, in our lab, we use fruit flies to study collective cell movements. If you don't know already, fruit flies rock. They live fast and die young, so we can get many different samples. They are easy to genetically and pharmacologically manipulate, as you see with all of these different pretty phenotypes here. And importantly for this talk, the embryos of fruit flies are optically clear, which allows us to get very nice high-resolution videos of embryonic development as it happens. But then there is this challenge of what do we do with all of this video data that we acquire? Well, we aren't just interested in studying these processes qualitatively, but in fact quantitatively, and that's because quantification allows us to detect more nuanced phenotypes, create more reproducible results, and really just get the most out of our data because we can make many different measurements from one data set. So for example, here is a developmental process that we study in our lab, which is heart tube formation. And what you're seeing here are the nuclei of cardiac progenitor cells, which start on either side of the embryo and then migrate towards each other to form the heart tube. And if we want to measure something from this video, we first have to segment out each of those nuclei, and then we can make many different measurements, such as the centroid position over time, which is what you see here, fluorescence intensity, cell shape measurements, et cetera, et cetera. But I sort of skipped over that big step, which is this segmentation. And the segmentation involves essentially drawing the outline of each of the objects that you want to measure. So for example, in a video like this, where you're tracking maybe 30 nuclei over 200 time points, that's about 6,000 circles that you have to draw for one video. And for a full experiment of 15 videos, you're looking at about 100,000 circles that you have to draw by hand. And so obviously, we want to be able to automate this process. And there exist many image processing algorithms that attempt to do so, such as the watershed algorithm. However, often these algorithms don't extend well to new data, and they're inaccurate. And this is a big issue for performing research because we really need there to be very high levels of accuracy. Any errors that are made, we can't just leave, we have to go back and correct because then we could be publishing erroneous results. And so neural networks are an obvious tool to try to alleviate some of these issues because they can be trained on problem-specific data, making them both more generalizable to new tasks and also more accurate. And a very common network architecture for segmentation is the U-Net. The goal of the U-Net is to take an input image, feed it through the network, and get the segmentation of the desired object as the output. And in this diagram, each of these rectangles is essentially a functional block of the network. As we go down the left-hand side, we have the encoder of the network, and this essentially pulls out the image features important for segmentation. And then as we go up the right-hand side, we have the decoder, and this pools together those features and assembles them into the segmentation mask. Now, UNets perform quite well on bright, well-defined images, and importantly, on very small data sets. However, they don't extend well to video data. And that's because UNets are unable to use that temporal information that is abundant and important in videos. Now, one way of trying to add that temporal information is to add recurrence within network layers. And so in this case, each layer in the network is seeing both the input as well as its own output from the previous time point. And an example of that is in long short-term memory layers, or LSTM layers, which you see a diagram of here. And you don't need to worry about the details. What's important is that this layer sees an input, its own output from the previous time point, and then separately maintains a memory of what has happened many time points prior and what it thinks is important to remember. Now, the issue with LSTMs is that they can memorize erroneous information, leading them to make the same errors on many frames. And then for a scientist annotating a video with one of these networks, that means that they then need to go through and manually correct all of those errors. And so a different way to try to add recurrence is to do this at the network level and perform semi-automatic segmentation by taking advantage of promptable neural networks, such as the segment anything model. In the segment anything model, or SAM, there is an image encoder to process the image data, but there is also a separate prompt encoder that allows the user to feed the network points or a bounding box or a mask defining an object to tell the network what to segment. And so you could imagine using this network in a recurrent way where you just take the output from the network from the last time point and use that to prompt the network for the current time point. And then the user can go frame by frame through the video, segmenting using the network's previous outputs or correcting errors when necessary to stop the propagation of those errors as they happen. Now, SAM is a large transformer-based model originally trained on over 1 billion natural images. And there also exists a version of SAM called microsam, which has been retrained on microscopy images. However, both SAM and microsam require a large amount of training data. So for scientists who are working on specialized segmentation tasks, it's simply not feasible to try to retrain these networks. And so that's why we created ReSCU Nets, which incorporate this recurrent prompting at the network level, but are built off of a UNet backbone to take advantage of the really low data requirements of convolutional neural networks. So what you see here in the diagram is that UNet backbone that you saw before in light blue. And then in dark blue, we have added an additional encoder that takes the mask from the previous time point, processes that for some amount of encoder blocks, and then combines that with the input image stream. And actually, in some design decisions that I am not going to show you today, we found out that just having this very first encoder block is sufficient to incorporate that temporal information. And so by increasing the number of parameters only by a few thousand, you're able to very nicely incorporate information from previous time points in a semi-automated way. And we have implemented these networks in our lab's Python-based image analysis software, Pajamas, which I will come back to promote later. And in order to test these networks, we also assembled 3 different microscopy video data sets, all of which were acquired in our lab. The first one is those cardioblast nuclei that you saw before. And what I want to point out here is that we don't always want to segment every single object, because not every object is going to be measurable. So for example, in this video, we have nuclei that move out of the frame, and also we have these dimmer nuclei here, or nuclei lagging behind the leading edge, and both of these are indicative of different cell fates that we don't care about, and so we do not want to segment or measure. Another system that we study in the lab is epithelial wound healing, and so we create a wound, and then we want to segment out the wound area, and this is a very difficult segmentation task For starters, because our label for cell-cell outlines is kind of crappy, and so it photobleaches very quickly, but also because the wound edge gets occluded and also cell-cell adhesions are removed from the wound edge, leading to a discontinuous border of this wound. And so this is a segmentation task that many networks actually have a very hard time performing well on, despite the fact that it appears quite easy to the human eye. And finally, we wanted to test whether networks can segment tightly packed objects. And so we acquired a data set with a nicer fluorescent marker that allowed us to generate ground truth masks of the cells around those epithelial wounds. And we compared our ReSCU Nets to different segmentation workflows, including UNets and the automatically self-configuring NN UNets, which both take a single image frame at a time, LSTM UNets, which take an entire video at a time, and SAM, which is a more direct comparison, which we're using in this recurrent prompting semi-automated way, where given the segmentation of the first time point, the network can then work off of its own outputs to frame-by-frame segment an entire video, with the option for human-in-the-loop error correction. So first, applying these networks to the cardioblast nuclei dataset, we measured the IOU, or intersection over union, which is a measure of segmentation accuracy, but we also wanted to get an idea of how much time or effort it actually takes a user to segment a video with one of these networks. And so to do that, we sorted each of the objects into either false negatives or objects that we wanted to segment but the network didn't accurately segment, false positives or objects that we didn't want to segment and the network segmented anyways, and true positives, and this is really the measure of all of those objects that we wanted to segment and the network was able to do without user intervention. And what you'll see in these videos is we have labeled the correctly segmented pixels in white and false foreground or extra pixels that were added by the network in cyan and false background or pixels that were missed by the network in magenta. Now, what we can see pretty obviously right off the bat is that in UNets, LSTM UNets, and NN UNets, despite the fact that their segmentations are accurate, there are a lot of these extra objects that are added, lots of false positives. And so for a user segmenting this video with one of these networks, they would have to go through and delete all of these objects that they don't want. And that's because the network doesn't use the prompts from the user to know what to segment. Both SAM and ReSCU Net remove these extra segmentations, and ReSCU Nets, because they were able to be trained specifically on this data set, they are able to much more accurately and consistently segment out these nuclei as they migrate towards each other. Moving on to the wound margin data set, what I really want to point out with this data set is just how consistently ReSCU Nets can segment these difficult to segment objects. And you can see that in the spread of the accuracies. All of these other networks can once in a while get really nice segmentations, but they also once in a while get really poor segmentations. And that means that a user has to go through these segmentations with a fine-tooth comb to find those errors. ReSCU Nets, however, are once again able to accurately and consistently segment out these wound areas, leading to almost 100% of frames that don't need any error correction. And finally, perhaps the most dramatic data set is these cells around the wound. So once again, we see that in those non-prompted networks, we have lots of false positives, and perhaps maybe unexpectedly, also many false negatives. So they're adding cells that shouldn't be there and removing cells that should be there. SAM is able to get all of the cells that you want. However, you can see, especially demonstrated in this video, those segmentations aren't consistent over time because, again, it wasn't trained on this problem-specific data. ReSCU Nets, however, are able to, again, accurately and consistently grab these cells, and we can see that segmenting a video with a ReSCU Net would require much less effort compared to any of these other networks. And so just briefly, I've then gone on to use these networks to enable some of my own research. So I study embryonic wound repair in the lab, which means that I get to say that I shoot fruit fly embryos with lasers as my job. And that's exactly what we do. We take a high-power UV laser, we ablate 4 or 5 cells in the embryonic epidermis, and then we study how the cells around that wound work together to close over that wound area. And to do so, they have to coordinate the polarization of myosin, which is a molecular motor, and myosin creates a cable around the wound that generates tension to pull that wound closed. And I was interested in understanding how these cells are actually able to signal to each other to coordinate this behavior. Now, calcium waves are, in many systems of wound healing, including our own, the first signal that occurs upon wounding. And they are important for telling the cells that the wound is there and in order to start coordinating their responses. So, for example, in the pupal nodum, which is just another tissue later on in the fruit fly life cycle, there is this large calcium wave that extends several cell rows away from the wound immediately upon wounding. And the role of this calcium wave sort of makes sense because we know that cells farther away from the wound also participate in this wound healing response and the reorganization of the cytoskeleton. In our system, we also see this calcium wave that extends several cell rows away, but we know that only those cells that are immediately adjacent to the wound seem to be participating in the wound healing response. And so the question is, why do we need to propagate this calcium wave to those distant cells when they don't seem to be participating? And to do this, I've been looking at gap junctions, which are the transmembrane structure responsible for the propagation of that wave to the more distant cells. And what I noticed when I blocked gap junctions with the pharmacological gap junction inhibitor carbinoxolone is that, first of all, wounds close more slowly, and we can segment out the wounds to get wound area over time, fit an exponential decay to that, and extract a closure rate constant. And we see that, indeed, when we block the propagation of this calcium wave, embryos have trouble closing these wounds. Now, what's happening to those more distant cells? Well, I used ReSCU Nets to segment out the cells around the wounds under this treatment. And what we notice, first of all, in controls, is that these cells that are positioned dorsiventral to the wound or at the top and bottom of the wound in this video, they begin elongated towards the wound direction. And then as wound healing progresses, they actually elongate even more dramatically to sort of cover over that tissue gap. And we can quantify this elongation and look at the change in elongation from beginning to end, and we see that even in multiple cell rows away from the wound, there is this significant elongation towards the wound area. However, when we block the propagation of that calcium wave by blocking gap junctions, we completely lose that elongation both in those first row cells where we know they're participating in the healing response, but also in cells several cell rows away. And so this analysis, which was enabled by ReSCU Nets, suggests to us that the calcium wave is important for signaling to those more distant cells to undergo cell shape changes important for facilitating wound repair. And now, just in the last few minutes of my talk, I want to promo some of the tools that our lab develops. The first is Pajamas, which I briefly showed earlier. Pajamas is an open source Python-based image analysis software, and it allows users to open, explore, manipulate, segment, and measure their images. There are some really nice tools in Pajamas, including we have a notebook system, so you can go from your experiment data set to notebooks that have a bunch of measurements and visualizations. You can apply convolutional neural nets like UNets or ReSCU Nets entirely through the graphic user interface. There are other segmentation approaches such as linear classifiers and watershed. And maybe for this audience, we also have a plugin system, so it's really easy to go from Python code and create a plugin for Pajamas and create your own image processing tools and pipelines in the Pajamas software. We also develop another tool called Junkie, or a Jupyter Notebook Image Explorer. So this is a widget-based tool that allows you to explore images in Jupyter Notebooks. If you don't work with multidimensional images in Jupyter Notebooks, it's kind of annoying. because you have to, if you're just using matplotlib imshow, you choose a slice of your image, you imshow that, and then you have to run the cell again with a different frame number in order to look at a different slice. And so it's really hard to sort of explore your images interactively in Jupyter Notebooks. And so that's why we created Junkie, which allows you to choose an image either from a NumPy array or from a folder, select the channels of the image, and then you can go through and slide through the channels, the time points, the Z slices. You can change the contrast, you can change the color map, none of which you can see on this example image that my PI decided to use to create this GIF. But trust me, it's a really convenient and useful tool. But I played around with this for a bit, and I thought, okay, this is nice, but usually when I am looking at images in a Jupyter Notebook, it's because I'm trying to prototype some sort of image processing pipeline. And so if I wanted to use Junkie, and I have this parameter space of the pipeline that I want to figure out, then I would have to still rerun this cell every time I change a parameter in order to see what happens to the image. And so I created a simple extension to Junkie, and I called it Junkie Picasso, which allows you to define any sort of abstract function that takes in an image and outputs an image. And so this is very small, and I apologize, but this is an image that a function that takes an image, some offset that it will add, and a sigma to perform Gaussian blur. And then by giving that function to Junkie Picasso, it gives you more interactive widgets so that you can play around with all of those arguments to that function and sort of interactively explore the parameter space of that function and see what it does to your image. And so sort of to summarize what I've shown you today, by adding network level recurrence, we enable semi-automated multidimensional segmentation, and these ReSCU Nets outperform state-of-the-art models on microscopy video segmentation tasks. And then I have used ReSCU Nets to show that gap junctions are required for cell shape changes that facilitate rapid embryonic wound healing. And with that, I would like to thank my lab. My lab mates have been super supportive, despite the fact that they all come from biology backgrounds. They have helped me immensely in acquiring the data and testing out the tools that I have developed. And I will just leave these up here in case you are interested in any of the tools, and I'm happy to answer any questions that you may have.
As a reminder, please ask your questions via the Slack channel. The Slack channel is track-bioinformatics, and we have a few questions already there. So Beth asks, were other common microscopy segmenters like CellPose or Stardust tried?
Yeah, so we have, I haven't formally compared my model to CellPose or Stardust, but we have sort of tried them on our own. And so the issue with Stardust is that, for starters, it isn't, as far as I know, implemented into any software. And so ReSCU Nets we have implemented ourselves, and it was very easy to put them into our software since they're sort of a very simple extension of UNets. But on our own data, I actually have not had any success using Stardust. CellPose, at least at the time that I tried it, didn't have good support for multidimensional data, and so it was quite difficult to go through videos and segment with CellPose. I know that they have come out with a couple of updates since then, so I'm actually not sure if that situation has improved.
Sneakers the Rat asks, loves the focus on small data, big fan. Could this approach be adapted to genetically encoded calcium imaging of neurons, where the positions of the neurons are static after stabilization, and segmentation depends on recognizing regions that have dynamics that match some canonical temporal pattern or kernel?
Interesting. Okay, so if I understand the question correctly, it's about, will the network be able to look at a cell that has some sort of specific temporal information and understand that that is that cell and to segment it? in which case, I think yes. You would need to, so currently ReSCU Nets only look at the immediate previous time point, and that's because for most segmentation tasks that we have, if we're imaging with a good enough temporal resolution, you really only need to see where the object was just before you took the current image in order to know where the object will be. I believe that if you gave it more temporal information, the network is quite good at recognizing which objects are which. And so potentially, if that temporal information is complex, then the network would be able to sort of use that to tell which cell is which, if that was the question.
Ian asks, does your network maintain the identity of the segmented object between frames in a time series? And if so, how?
Yeah, so we're actually segmenting object by object. So when we prompt the network, we give the network the mask of a specific cell, not all of the cells in the previous time point. And so that means that tracking is essentially built into the network because it will segment what it thinks that cell is in the next time point. And I will say that at least in my own experience, I have never had an issue where I give it one cell and then it actually segments a different cell that's nearby. As far as I've been able to tell, it has 100% accuracy in segmenting things. So that also removes another step, or not segmenting, in tracking things. So that removes another step in the pipeline. You don't have to put this through another tracking algorithm.
Roberto asks a software question. Are there any challenges of distribution or packaging that this type of software faces? How easy is it for other users to install and use the package, and what are the pain points? How is it for other algorithms in the field?
Yeah, so this is sort of a big issue for us. We've had a lot of challenges distributing pajamas specifically. We use TensorFlow in pajamas, and I don't know if anyone has solutions for this, but TensorFlow has been a big pain in our butt trying to distribute pajamas over several OSs and versions. I have rewritten all of our code into PyTorch. However, that was a significant change, and so that's going to take a while before my PI can go through that and make sure he's comfortable with everything. But in my experience, and I think in other available software, PyTorch is much easier to version compared to TensorFlow.
So with that, let's please give a round of applause to Ray. Thank you.
Thank you.